Hello, everybody. Welcome back. This colloquium has thus far been unbelievably on schedule. We will try not to get too out of hand as we go forward. There will be a slight change tomorrow. One of the speakers canceled. There was one added speaker. We'll have more information about that by Susan Marty at the end of today. She will have handouts with the new schedule for tomorrow. One of the things that'll make you happy is that we end at 4:30 tomorrow instead of 5. But so anyways, let's immediately turn to the speakers today. And the first one will be Elizabeth Iorns.
Great, thank you. Really happy to be here, and thank you so much for the invitation to talk today. And today I wanted to talk about replication of biomedical studies. And I'm here from Science Exchange. My background is actually a breast cancer biologist by training, and then I left academia to start my company Science Exchange about 5 years ago. So today I wanted to talk about why should we replicate studies, how to replicate studies, what replication studies have been conducted, and what are the key lessons that we've learned so far. So the first part is why should we replicate studies? So this is just from Wikipedia, the scientific method, and one of the key points that I'm highlighting is that really at the core of the scientific method is that the steps must be repeatable. So in other words, we need to be able to replicate those steps, and that's how we basically think about doing science. And then this slide really shows for reproducibility, this is a definition that we use for determining whether or not a result is reproducible, to replicate the experiment and see a similar finding. However, what I've found is that most scientists would agree that reproducibility is very important, but they assume that self-correction occurs through other labs just naturally doing similar experiments and publishing those results. And so this is a headline that was published in The Economist, which talks about, to an alarming degree, this reliance on self-correction is not really working. And this is some of the results that are available so far. So there are actually very few studies that have been conducted in the fields of biomedical research about the rates of reproducibility. So this is something that has been talked about enormously. We have all of these symposia, there's many conferences that are organized to discuss this issue, and yet we actually have very few data sets to really look at whether or not there is an issue with reproducibility and be able to actually use those data sets to understand what's associated with reproducibility. So these are some of the studies that have been published. They're not actually all studies. Some of them are just commentaries with no data, but they do give us an idea that there may be a problem with reproducibility and also with this idea that self-correction occurs. So for many of these commentaries, they actually correlated things like citations and impact factor with whether they could reproduce the results, and they found no correlation. So some of the core metrics that we think are associated with quality may not actually be associated with quality. And this is a survey that was conducted at MD Anderson about people's experience with reproducibility. So here they found that 50% of respondents had experienced at least one episode of not being able to reproduce published data. But I thought that this fact was actually even more interesting. Of those respondents that could not reproduce data, less than 30% of them actually published that they could not reproduce that data. So this is where that issue around assuming that self-correction occurs comes from. So why don't scientists publish their failures and enable self-correction? Because we don't have a culture of replication. Their replication experiments are not the norm. They're actually, as we saw, very unusual and very infrequently reported. There are issues with the academic culture around retaliation, about publishing failed replications. and they can be difficult to interpret the results, particularly from studies that haven't been formally designed as replication studies. And so these create a really poor incentive for actually doing these studies and publishing these studies. And that ultimately means that the literature does not self-correct. So why should we replicate studies? Because many studies may be irreproducible based on the limited data sets that we have. And replication studies enable the identification of reproducible results. By definition, they are the mechanism that we use to determine whether a result is reproducible. Designed replication studies are very rarely funded, conducted, or published. In the US so far, for biomedical science, there are only 2 biomedical replication attempts that have been published, one of which was funded by the Laura and John Arnold Foundation, and one of which was a small study funded by the NIH. So this means that we don't have a mechanism for correction. So how do we replicate studies? How we've thought about replicating studies is to do direct replications. The reason why we want to do direct replications was actually explained quite well this morning by one of the earlier speakers who talked about the idea of being able to generalize results, but the first step is actually replicating that to check whether the direct result is replicable. So we're doing direct replications, and this allows us to then take that result and do extension studies to see whether the result is generalizable. Where possible, we obtained input from the original authors, as well as unique non-commercial reagents, which in biomedical science actually make up a fairly large proportion of the types of reagents that are used, as well as data sets. We pre-established and pre-registered protocols and analysis plans for replication studies that are being conducted. We use power calculations to ensure appropriate sample size. We use expert independent labs with extensive expertise in the techniques being replicated. And we published all protocols, results, and data so that our replication publications were themselves much more easily to take for reproducibility tests later on. So our goal was to create something that could be taken and reproduced in somebody else's lab with minimal effort. So the expert independent labs we used and how I'm involved with the project is through Science Exchange. So Science Exchange is a marketplace of scientific service providers, and these are more than 3,000 individual qualified contract research organizations, manufacturers, academic core facilities, and government facilities. And what's really important about this network is that it's highly scalable. We can easily identify a particular expertise within a searchable database, and we can work with that lab immediately because they've already been pre-qualified and pre-contracted. And that scalability was very important for being able to do these experiments cost-effectively and quickly. So what replication studies have been conducted? These are the projects that Science Exchange has been involved with, and they do form a pretty large basis of what has actually been published in this field. So these make up a broad type of different sets of analyses. There's the Reproducibility Project Cancer Biology, which I'm going to talk about the results going forward in this session. We've also done studies for the Prostate Cancer Foundation. There's the Reproducibility Initiative and Antibody Validation Initiative to look at the quality of reagents that are used, a reanalysis study that was funded by the Gates Foundation, and then private studies that are run frequently for pharmaceutical companies who are the ones that raise this as a significant challenge to their ability to take published results and be able to build on them. So I'm going to spend now a big section of the rest of my time on the Reproducibility Project Cancer Biology. This project is a collaboration between the Center for Open Science and Science Exchange, and its goal is to replicate the key experimental results. We started off wanting to do 50 top cancer biology studies as defined by impact in the field. And we defined impact in the field as number of citations as well as a combination with alt metrics like Mendeley readership and other types of leading indicators that were associated with citations going forward. So this is the progress of the study. These studies were selected. They were from the field of cancer biology from the years 2010 to 2012. So far, we've had 31 registered reports submitted to eLife. 29 have been accepted and published, and 2 have been rejected. 8 replication studies have been completed fully, and of these, 5 are published and available on eLife now. So I'm going to talk through those results since they're already published. And then we have 3 that are also complete that are in review right now at eLife. Of the remainder, we have 21 replication studies that are currently ongoing in labs, and we have 19 potential studies that are currently on hold. So these are the first 5 papers that have been published, and I strongly encourage you to go and look at these studies in eLife. We're really proud of the work that went into these studies, and I think they're really interesting when you really dig into the data that's there and the format and all of the work that eLife put into reviewing them and presenting them in a way that makes them, I think, very usable. So here, the first study was the BET bromodomain inhibition as a therapeutic strategy for cMIC. And I'm not going to talk through a lot about the individual data, but just to give you a sense for what does the original paper's data look like versus the replication studies that we obtained. So this study was run by the Developmental Therapeutics Core at Northwestern University. And this core facility is actually responsible for running the efficacy and toxicity studies at Northwestern University for investigators who have preclinical programs that are going into clinical studies there. So they have enormous experience with animal models, with running the types of experiments that were conducted in this study. The cost of the study was $39,200, and the timeline was 5.8 months for the experimental work. So this is the first result where we're looking at the effect of a drug on the expression of MYC. And you can see in the original figure that when you treat them with the drug, there's a decrease in the expression of MYC. And we saw the same thing in the replication study. We also treated xenograft modeled animals with this drug, and in the original study, they saw that the drug resulted in increased overall survival. We also saw a statistically significant effect on increased overall survival. And we also measured tumor burden based on an IVUS assay, and in this case, we did not see a statistically significant effect. The last piece that you can really dig into in the papers, which we're trying to get at, what does that data mean when you combine it with the original data set and the replication data set? So here we did a meta-analysis that combined the 2 different data sets. I think there's a lot of really interesting approaches that we heard today that I'm excited to collaborate with people on, and the data is all available for anybody to do any types of analyses that they want. So we're very interested in other people taking the data and doing things with it as well. The next study is the Sugihara study. This was co-administration of a tumor-penetrating peptide to enhance the efficacy of cancer drugs. And again, this study was run by the Developmental Therapeutics Core Facility at Northwestern University. The cost of the study was $43,500 and took 5 months. So here, the original study reported this peptide was able to increase specifically tumor-specific accumulation of DOX in the animal model. And in the replication study, we did not observe this increased tumor-specific accumulation. And then this study was looking at the tumor weight following treatment with either DOX alone or with the peptide that was aimed to increase the tumor-specific penetration of the DOX. And so here you can see that in the original study, the DOX alone had a small effect, and then when you combine it with the peptide, you see a significantly increased effect on the tumor weight, which was not observed in the replication study. We also monitored body weight of the mice over time, and our replication study just looked at the lower dose of DOX, which showed very similar results to the original study, where there was no real effect of DOX or the peptide on body weight of mice. And tunnel staining was performed to look at apoptosis in the tumors. And here, the original study found that there was an increase in apoptosis measured by tunnel staining when there's addition of the peptide. And we also did not observe that in our replication study. So again, we did a meta-analysis, and this shows the original effect, the replication, and then the meta-analysis of each of the effects that were reported in the replication study. The next paper is the discovery and preclinical validation of drug indications using a compendia of public gene expression data. In this paper, a statistical approach was used to analyze data sets and come up with predictions about whether drugs would be effective in particular xenograft models. Again, this work was done by the Developmental Therapeutics Core Facility at Northwestern University. The study cost $11,800 and took 4 months. So here we can see that in the original study, they predicted that a new drug would basically be effective against one type of xenograft model but not another, and that's what they saw from their predictions, and it's a little hard to see. So the doxorubicin is the control, and then on the replication study you can see the vehicle versus the test drug in the middle, and we tried to use the same colors so that you can compare, but basically they are very similar results. And then no effect was observed with this drug on the control tumors. So again, very similar results are observed in the replication versus the original study. And a meta-analysis was performed, and so this again shows that when you combine the effects, you can really start to look at what is the certainty once you've got two different studies that you can combine together. This study is the CD47 signal regulated protein alpha interaction as a therapeutic target for human solid tumors. This study was run by Noble Life Sciences, which is a contract research organization. This organization has many accreditations, including ALEC accreditation. It's GLP certified, has been inspected by the FDA, and has submitted hundreds of studies for IND-enabling projects that have gone into the clinic. So they also have a lot of experience with doing animal models to examine toxicity and efficacy of potential therapeutic compounds. This study cost $11,700 and took 8.5 months. So the first analysis that was replicated here was the effect of the test article on blood toxicity results, and the replication presents the results slightly differently, but you can see the key ones to look at are these results here. So in these three results, they're looking at red blood cell count, hemoglobin, and hematocrit, and in all three of those analyses in the replication studies, we did see a significant impact. This was also observed in the original study. And the next part of this replication was to look at tumor weight of immune-competent hosts treated with control or with the therapeutic test article. And the test article that we looked at was the most effective antibody here, which is the MIAP410. So here we see that there was no effect in the replication study versus a very significant effect in the original study. And this just shows the meta-analysis when you combine both of those two different data sets. And the last of the 5 original studies that have come out is the melanoma genome sequencing reveals frequent PREX2 mutations. So this was also run by Noble Life Sciences. It cost $29,700 and took 10 months. This study required use of a partially transformed melanoma cell line, and this basically was then, we then expressed different PREX isoforms in the cell line to look at their effect at being able to fully transform the cells in animal models. So this just shows that these new versions of the cell line have been made with the different mutations. And then their effect on tumor-free survival was measured in a xenograft model. And so in the original study, the different mutations that we looked at in the replication are spread across 3 different graphs, but they're both the most significant effects. So those really in the original study had a significant increase in the time at which the tumours were able to develop. So basically, tumour-free survival was significantly decreased when you transform the cells with these PREX2 mutated isoforms. In the replication study, we found that the cell line had become fully transformed over time, which is a common occurrence in cell culture, and so we could not detect any effects of these different isoforms. So we did do also a meta-analysis for this, but I would say that the results were confounded by this inability to see any effect because of the transformation of the cell line. So in summary, the first of the 5 RPCB projects that have come out, we obtained a very broad spectrum of results. It was really interesting to look at them and just see what happens when you try and replicate results in another lab in biomedical sciences. And so of them, the first results that we've seen is that 2 have obtained actually pretty similar results to the original studies, and 3 obtained different results to the original studies, and those could be for a variety of reasons. The average cost per study of these 5 studies was $27,200, and the average time to conduct the experimental work was 6.7 months. So the next part of my talk, I wanted to talk about the key lessons that we've learned so far from conducting these studies. So the first lesson, which I think we've heard a lot about already in the symposium, is that publications often have insufficient documentation to enable replication or follow-up studies without contacting the original author. And then when you contact the original author, this actually is quite problematic because you are often faced with a situation where the person who conducted the work has left the lab, and this occurs very frequently and very quickly. So there's a rapid loss of the ability to really get access to anything that was conducted in terms of protocols, reagents, or data, and this decay is really difficult to actually take the published result and be able to do a direct replication or to build upon it as efficiently as if all of the information was there up front. We also found that reagents are often not uniquely identified in the publications. They're also often not available and cannot be easily shared due to IP requirements of universities. So when somebody generates a new animal model at a university, there's actually IP restrictions around being able to transfer that out of the university. And so this creates additional challenges with being able to access the reagents that have been created and are not commercially available. We also found that raw data is infrequently stored or available, and this is something that I think has started to be addressed since that time period was from 2010 to 2012. I think there has been progress made in making many more raw data sets available with publications, but we should still really emphasize that this is a key area that we could improve upon very easily. And then the 2 last points are that one of the big criticisms of this project was that replications would be very costly and that they would not be practical. Some of the original quotes were things like, "This would double the NIH budget." And I think it's clear that that's not true. So there are ways to do these studies very cost effectively, and it's not necessary to replicate every single experiment that's ever done, but you can replicate the key experimental results for really important studies that other people are going to be trying to replicate in industry labs and in other academic labs, and that you can make those studies available so that people can learn from other people's experiences. Replications are difficult, and it has been a significant learning curve in how to conduct replication studies. They also frequently generate criticism. So this is something that I think ultimately falls into that category of why are these studies not done or published? A big part of it is that there's very poor incentives to actually do these studies. So these are some of the data that we've collected about some of those points from the Reproducibility project. So one of the things we looked at was author responsiveness. As I mentioned, we did contact all of the original authors that were part of the data set that was selected, and we asked them for input because in all cases there really was not sufficient information in the protocols to be able to do a direct replication. We were able to reach 62% of the authors, so we had 31 of them respond to us. Of those 31, the timeline for responsiveness was highly variable and did cause significant delays in being able to move forward with the replications. So here you can see the mean time was 67 days with a max of 354 days. But of course, this is only the authors that we had a response from at that time. So you could actually extend this out to be infinitely long in theory. So that author responsiveness issue comes back to we need to have a system for having the information that you need available at the time of publication, because if you have to rely on going back, then very quickly you're missing so much of those key pieces that allow you to build upon the work. We also looked at author supportiveness of the study, and in general I think that the authors were quite supportive of the study. You can see here that about half of them were characterized as either positive or enthusiastic, and of those there was no correlation between their supportiveness and the time at which they responded to us. So it's not about whether people are supportive of doing replication so they're more likely to respond to you. It's just that it's really challenging. People don't have a way to just quickly get access to the things that are being requested of them, and it's a lot of work. And so then it takes them a significant investment in time and effort themselves to be able to respond to these requests. We also did a project with the Resource Identification Initiative to look at the ability to uniquely identify reagents that were in the original published studies. And you can see here the fraction that were uniquely identifiable. For most of the different types of reagents, less than 50% were uniquely identifiable. So again, that comes back to having to go back to the original lab. So ask them, okay, well, if a p53 antibody from cell signaling was used, which p53 antibody was used? Because we can't uniquely identify which one. And this lesson around the cost, this is something that I think is incredibly interesting because it was one of the biggest criticisms. And actually this came out recently from the former EVP of research at Merck. And he talked about the cost of having to try to replicate experimental results that are published in industry labs. And so you can see here, he talked about it takes approximately 2 to 6 scientific personnel, 1 to 2 years work in an industry laboratory to try to reproduce the original investigations, and their cost estimates average between $500,000 and $2 million. And when we publish a really exciting breakthrough result, all of these industry labs are taking those results and hoping that they can build upon them to develop a new therapy that they can bring into the market. And all of them are doing that individually, and none of them are publishing their findings. They're publishing the summary findings occasionally, like we saw in the reports from Bayer and Amgen. But this work to try and build upon published results is also happening at academic labs, and they're also not publishing that. And so there really is a big disconnect there between the value associated with doing replication studies to just put more information out about the potential of a result to be able to be reproduced versus the costs that are currently being spent already on trying to build upon these results. So some of our suggestions for improvements are to reduce the reliance on contacting original authors. So this is, again, not related to authors not wanting to be helpful or not wanting to be collaborative. It's just down to not providing them with any tools to be able to do this effectively. And I think there's a lot of steps that have been made that we can really leverage in this process. One of those tools which we actually use for the reproducibility project is the Open Science Framework. This is a free tool that you can use where you can register all of your studies, you can track all of your experiments that are going on, and it can all be done in a private collaboration and then just made public when you want to link to it. And we used that repository for all of the projects, and they're linked to it in the eLife publications. There's also a lot of automation that's happening now that I think is really exciting, and this automation can definitely help with being able to have a complete capture of data from a machine all the way through to a data set that's then analyzed. We talked already about the ability to identify commercial reagents. This is something that the Resource Identification Initiative is working on, and they have partnerships with many publishers now. And then one of the last points is repositories for materials. This is something that's often quite underfunded, but very impactful for the research community. So these are repositories like AdGene, Jackson Labs, ATCC, and they're where people can store animal models, cell lines, and plasmids, and then they're readily accessible to the rest of the research community. That means that it's much more easy to be able to build upon those results that are published using those reagents, and also it makes it much more easy to do a replication with those particular materials. Somebody else actually already talked about this suggestion today, but I think it's really important to look at the types of studies that we're doing and categorize them correctly into whether they're exploratory studies or confirmatory studies. This is something that is infrequently done, and when you look at our structure by which we fund science, a lot of it is based on exploratory novel studies, and our grant mechanisms exist and support these studies fairly well. But confirmatory studies are different. They do require technical experts. They do need to be milestone driven, and they may require a different type of facility to be able to run them effectively. And the last suggestion is to implement QC measures for these confirmatory studies. So there's definitely been some proposals from the community around introducing more generally a GLP-light type of infrastructure for more basic research. And I think that proposal is probably best suited for something like the confirmatory study space, because you can do things like you can pre-register the experiments, you can power them because you have an effect size to work off, and you can do things like systematically control for bias because you have all of the trained investigators and the personnel are staffed appropriately to be able to do those types of studies. And my last suggestion is to provide funding for replication studies. Recently, this was the first moderate amount of funding that was provided by the federal government in the Netherlands to fund a replication program. And certainly, I think, based on the enormous interest in this issue, we see a lack of funding to actually do these types of studies to be able to learn from them. And given that these studies are how we are actually able to assess reproducibility, this is, I think, a challenge. Our studies were funded by the Laura and John Arnold Foundation, which is not a public biomedical funder. I definitely think there's a role for the public funders to provide funding for these studies in order for us to learn whether any of the other interventions that are being proposed are actually effective. Because right now we don't have a baseline and we won't have any way to track whether the things that we do have an impact on our ability to improve reproducibility going forward. So thank you very much for the opportunity to speak. I really do appreciate it. I would like to thank many of the people who conducted this work. So the RPCB core team has put in an enormous amount of work to get these studies off the ground, and now the project is just rolling through and being completed. I'd also like to thank the authors from the selected studies, who many of which were very supportive and had to put in an enormous amount of work themselves in order to be able to provide data sets or reagents for the studies. I'd like to thank the Science Exchange labs that conducted these studies, the Center for Open Science, eLife, the Laura and John Arnold Foundation, and the reagent donors that helped support the project. Thank you.